you for inviting me. I'm very excited to speak with you all about some of the recent work that we've been doing in the lab, including this little story that we just published as a preprint, and then also how we're kind of building upon that from the drug discovery process. So, just as a little bit of background, of course, when it comes to innate immunity, we can think of this as acting on two levels. One is our adaptive immune system, which is composed of our specialized immune cells, such as our T cells and B cells. But at a cellular level, all of our cells have their own special immune systems, which we call the innate immune system. And that is specifically there to protect cells from the infection by pathogens that otherwise would try to invade and rewire host cell machinery to reproduce themselves. And so pathogens over millions of years of evolution have had to develop a number of different strategies for antagonizing these different responses, ultimately to create a fertile environment where they can reproduce themselves. And of course, over the past couple of years, the big pathogen that we've all been kind of looking at is, of course, SARS-CoV-2. We've been really interested, especially at UCSF through the AViD consortium, to understand what are the proteins made by the virus, what do they go on to do, whether they interact with host factors or rewire certain aspects of the host machinery to help reproduce the virus, and also very specifically to AViD, which targets should we be going after to develop therapeutics against, so that we have other strategies in combating the virus beyond vaccines. And so a lot of the work for the different main projects at AViD have been really focused on understanding what do these proteins do, what do they interact with, what do they rewire in the cell, and then also, of course, developing drugs against them. Of course, a lot of this has been underpinned by some of the work that was previously done at UCSF near the onset of the pandemic in 2020, which was captured in this really seminal paper, which was focused on mapping out the different interactions made by viral proteins in related coronaviruses, such as SARS, MERS, and SARS-CoV-2, and the host proteins that they interact with to understand what are the conserved interactions being made by viral proteins with their host factors to develop a greater understanding of which targets it would be best to go after. And while, of course, as I mentioned, there are a number of projects working on being embarked on at UCSF that are focusing on various different aspects of the viral machinery, what I've been specifically interested in through a developmental research project that is funded by AViD has been really looking at an accessory protein to SARS-CoV-2 and SARS-CoV-1, as well as some other related coronaviruses called ORF9b. And this has really been something that's fallen out of this paper right here from some of the really great structural work done by Clem Burba's lab, which is capturing this cryo-EM structure of this complex between TOM70, which is a mitochondrial outer membrane receptor involved in the import of cytoplasmically translated proteins to the mitochondrial inner matrix, and ORF9b right here, which is an accessory protein to SARS-CoV-1 and SARS-CoV, illustrated right here as this amphipathic helix that binds to TOM70, presumably what we think right here at this substrate recognition binding site. And so what's really interesting about this is that it kind of underpinned two interesting observations that we were really interested in exploring. And the first is kind of illustrating that the mitochondria, of course, beyond being this platform that's incredibly important for producing energy for cells, is also actually a platform for antiviral innate immune signaling, where a number of different antiviral proteins in the cell aggregate to induce antiviral responses and drive the transcription and activation of genes that are involved in activating an antiviral state. And specifically, we see that ORF9b co-localizes to the outer membrane of the mitochondria and interacts with TOM70 specifically. And through that interaction with TOM70, ORF9b is able to suppress the innate immune response through this type 1 interferon response. But what's also really interesting about this is that we actually knew what SARS-CoV-2 ORF9b looks like before what we knew the structure of ORF9b bound to TOM70 was, which is that we could see in the crystal structures of ORF9b that it crystallizes as this homodimer of beta sheets, where you have two chains of ORF9b illustrated in this gray chain and also this magenta and pink chain that come together to form the homodimer. And so presumably then what has to happen is that this area right here highlighted magenta, which forms a beta sheet, has to undergo some sort of refolding in order to bind to TOM70, as we see in this structure right here, which adopts this amphipathic helical fold. Now, again, these two structures were really interesting to us, where we have the structure of ORF9b bound to its host factor, but then also ORF9b crystallized on its own. And what we have been really interested in trying to understand is how exactly does this interaction come to be? How exactly does ORF9b go from existing in just one conformation in the oligomeric state, which is this homodimer of beta sheets, to this other conformation, which is this alpha helix that then binds to this host factor and is involved in driving the suppression of innate immunity? A lot of this has been guided by some of the previous work that's been published, which has illustrated that the homodimer specifically does not go and directly interact with the Tom70. So you don't get this encounter complex where separately purified ORF9b engages Tom70 on its own, and that in some way drives a conformational change and refolding that then results in the complex that we see in both cryo-EM and crystallography structures of the complex. What we thought had to happen is that if this doesn't directly interact with Tom70, what probably has to happen is that there has to be some sort of equilibrium within the cell between these different states. So an equilibrium between ORF9b monomers and homodimers, but where the where the monomers of ORF9b are able to go and specifically interact with Tom70 and drive the suppression of interferon that we see. And then also what we think is that this has to be regulated by the cell in some capacity, because what we can see from some work that was published by Nevin Krogan's lab is that ORF9b can be phosphorylated by a host kinase. And presumably, when that phosphorylation occurs, it blocks the interaction with Tom70 and results in an improved interferon restoration. But presumably, it also doesn't interact with or affect the ability for ORF9b to then to rehomodimerize itself. So in some capacity, the cell is in some way manipulating or regulating this equilibrium to some capacity that then results in a restoration of interferon when phosphorylated, but on its own can then go and bind to Tom70 and then drive the suppression of interferon. And this is really kind of the system that we've been really interested in trying to understand and work within, both to understand how this equilibrium between ORF9b and Tom70 is established, but then looking to see whether there's some way that we can bias this equilibrium, some way of basically recapitulating what we see with the phosphorylated ORF9b, but focusing in on the homodimer and trying to drive this equilibrium into a state where ORF9b is locked in a conformation that is then unable to interact with host factors like Tom70. And we've been really looking at the structures of ORF9b. We first focused in on what the structures of ORF9b could maybe tell us about how we might go about doing this. And what we saw is that in the structures of ORF9b, going back to SARS-CoV and SARS-CoV-2, is that again, it crystallizes as a homodimer primarily composed of beta sheets. But what's really fascinating is that in this homodimer conformation, there's this central channel that runs the length of the homodimer. It's about 22 angstroms in length. It's open at both ends. And within the central channel, we can see electron density that supports the placement of some sort of long linear ligand, which we thought might be either something that arises from the crystal conditions, such as PEG molecule, or something that looks like a lipid. And I'll kind of spoil this by saying that from some of the work that we've been doing and some previous work that's been published, we have strong evidence to think that this is actually a lipid that binds within the central channel. But the placement of this from perhaps a drug discovery perspective was really kind of advantageous because just by where this is located, we might think that the binding of these lipid-like molecules within the central channel right here may act in a great way to sort of stabilize the homodimer by acting as like a little staple that keeps the two halves together. And because we know that the homodimer itself doesn't directly interact with Tom70, perhaps this has been some way of biasing and stabilizing the ORF9b homodimer in its state, where it would then presumably not be able to dissociate into monomers that then bind to Tom70. And so what we were interested in looking to see is there some way of basically isolating ORF9b homodimers in a state where we can look at both the lipid-bound structure, but also the apo structure that we could then work with for future studies. And what we did is we developed some system for refolding the homodimer to eliminate and basically wash out any co-purifying lipids that bind to this channel. And what we could see is that we could recover the ORF9b homodimer, where in the folded state, we see these very strong density right here that supports the placement of some sort of PEG or lipid-like molecule. But in the refold condition, we don't really see that density. We see some diffuse peaks that are likely arising just from background noise or just bulk solvents. But from some collaboration that we did with Daniel Sweeney's group at the Gladstone Institute, we were able to do some mass spec analysis to see in the natively purified and refolded purified ORF9b what the identity of these lipid-like molecules might be that co-purify. And we think that they narrow down to either a palmitoyl or steroyl-like lipid, which is also actually very consistent with a mammalian expression system that we have, which suggests that lipid binding is not something that's just unique to E. coli expression systems, but is a feature of ORF9b that is captured in other cell expression systems that are closer to humans. And also, what was really important is that we could see that the lipid binding itself is not responsible for ORF9b dimerization. We can recover the ORF9b homodimer without the lipid. So it doesn't necessarily suggest that lipid binding is something that necessarily induces ORF9b homodimerization, but it might be something that is a result of the ORF9b homodimer forming. Now, this little bit that I'm going to talk about here is related to some work that we recently published, which is really our efforts to develop a greater understanding or a foundation that we can build upon for how this system is established between ORF9b homodimers, monomers, and TOM70 that we can then use and leverage for the discovery of small molecules that then bias ORF9b. And what we did in this paper is that we proposed an equilibrium for ORF9b and TOM70, where ORF9b and TOM70 are in equilibrium between the ORF9b homodimer, monomers that have to undergo some sort of conformational change to adopt the TOM70 binding competent conformation that we see, and then of course the subsequent TOM70 ORF9b complex. We took this proposed equilibrium and then we derived a computational model that is built out of ordinary differential equations that we use to describe this equilibrium and our assays that we use for measuring this equilibrium. And then to underpin the mathematical representation for the rates for how quickly these different binding reactions are occurring, we also developed surface plasmon resonance assays to identify the specific kinetic rates for these micro reactions right here. So for example, the binding of ORF9b to TOM70, the binding of ORF9b to ORF9b to get at the dimerization kinetics, as well as some additional SPR that we did that are specific to the assays that we developed in order to understand how we can then model these systems and then derive more quantitative information about how things such as lipid binding bias it. And of course, we feed all of this into our kinetic assay that we developed, which is based on fluorescence polarization to measure the displacement of a fluorescent probe from TOM70 upon competition with an unlabeled competitor, which could be either small molecules, peptides, or of course, the unlabeled ORF9b homodimer. And so in the way that this assay works is that we took ORF9b, we chopped off about 2/3 of the protein, and we use the structure to really guide us in terms of what portions of ORF9b we want to focus on for creating a fluorescent probe, which is again driven by the cryo-EM and crystallography structures of the complex. So as I mentioned, we cut off about 2/3 of the protein because it has a tendency to dimerize with itself when purified on its own. And we derived peptides of this magenta region right here, where we appended a fluorophore, which is fluorescein, the C terminus, which we can then incubate with TOM70 to create these fluorescent complexes. And so, of course, with fluorescence or fluorescence polarization, when you have something that's a very small in solution on its own, it tumbles a lot, it emits a lot of light, but it's not polarized. But when it binds to something that's much larger than it, you get much less tumbling in solution. You get much more polarized light that's then re-emitted, which you can then use as a direct readout for binding or displacement. And then, of course, the way we think this might work with the unlabeled ORF9b homodimer is that when we use the homodimer as the unlabeled competitor, that is then going to establish an equilibrium with TOM70. So that homodimer is going to go into an equilibrium with monomers, and those monomers can then, of course, either rebind with other monomers to create the homodimer, or bind to TOM70, pick off that fluorophore, and drive a decrease in polarization over time. And that kinetic signal that we derive from the polarization assay, we can then use our models to fit to get, again, quantitative information about how different factors may bias that. So we, again, are using fluorescence polarization assay. We chase in to our pre-equilibrated fluorescence complex, the unlabeled homodimer, and then we take our model that we derive, which is ordinary differential equations in Berkeley Madonna, and we fit that to our experimental data right here. And what was really exciting to us, which is that in this top port right here, which is the lipid-bound homodimer being used as the competitor, the competition is very, very slow over time. And so these little dots right here represent the different concentrations that we use being measured over time. And what we see is that as the concentration gets lower, you get this kind of lag phase, I would call it, where there's little competition that occurs initially at the outset, but then there's very slow competition that then occurs over time, which is over the course of hours. But then when we use the apohomodimer, which is again something that we isolate by refolding the protein to kick out the lipid, we see these very fast kinetics where there's a very fast timescale where orphan B is able to establish this equilibrium with monomers that then bind to Tom70 and kick off the fluorophore. And by applying our kinetic model to our experimental data, which are these black lines right here, we were able to fit our data and derive information about how the lipid binding to the homodimer specifically is stabilizing the orphan B homodimer. And so while we were able to experimentally derive the KD of the apo-orphan B homodimer, which is in the low single-digit micromolar, our model gave us the opportunity to then figure out what aspect of this equilibrium is being biased when the lipids are bound to the orphan B homodimer. And what we specifically focused on was modeling the dissociation rate of the orphan B homodimer to then develop an inference for what exactly the stabilized KD would be, which is that when orphan B is bound by lipids to the homodimer, the KD or the apparent KD is in the low single digit nanomolar. So basically, the lipid binding to the homodimer lowers the KD by approximately 100-fold to really tightly stabilize the homodimer. But it doesn't fully inhibit the ability for orphan B then to bind to Tom70, as we can see where it reaches equilibrium. It just slows the whole process down on the order of hours. And of course, there's a lot more. I'm not going to go into more in terms of how else, some of the other things that we were doing with this. But of course, if you're interested in this story and how we also develop other assays and variants for testing this, I, of course, would direct you to our preprint that we have available if you're interested in that. But as I mentioned, that a lot of this right here was basically the layers that we had to develop to understand how the system works, how this equilibrium between orphan B and Tom70 is established, so that we can then go about rationally develop small molecules that target specific conformations and states of orphan B to find ways of probing this equilibrium, both to learn a little bit more about the biology, but also potentially to learn if there's a way of biasing this to restore interferon signaling. And the way we've been going about that is because, of course, there is no existing chemical matter against the orphan B homodimer. We have also been very heavy into using crystallographic fragment screens against a number of different SARS-CoV-2 targets. Another big target, of course, within our lab that we've been going after is, of course, the macrodomain, NSP3. But we've also leveraged this technology to go after the homodimer of orphan B. And so we developed a crystallization system for the orphan B homodimer that we could then soak fragments against to develop some of the initial chemical matter starting points. And so we soaked about 400 compounds and fragments into our crystallization system. And these are the four major binding sites that we saw where we got these fragments to bind. And of course, because the homodimer is symmetric, these sites, for example, sites 1 and 2 are mirrors. They're biologically identical to each other, just like sites 3 and 4 are also biologically identical to each other near the channel entrance where we see the lipid bound. And then just to go a little deeper in terms of what we're seeing here, this again, binding site 1 is on the surface of the protein. It's composed of these two chains of orphan B that together form this binding interface. And see, these are some of the representative binding poses that we see where the fragments kind of fall into this little trough and are aligned within it. But what's also interesting is that we also saw some fragments binding near the lipid binding channel entrance at both ends. And we thought that this could be really exciting to further explore for our systems that we developed and some of the information that we developed from our previous paper that we published to see, is there basically some way of taking these compounds and improving upon them to target the lipid binding channel to basically mimic some aspect of lipid binding to either stabilize the homodimer in this state where it doesn't bind to Tom70, but also to get, again, at more of the basic biology of orphan B, because we don't really know what lipid binding does for orphan B. We don't know why it binds to lipids. We don't know what role that plays in viral infection, but having some sort of tool compound that occupies that channel maybe give us some way of developing an understanding of how lipid binding plays into SARS-CoV-2 infection, but also orphan B's function within that. And so again, we took our original hits right here that we could see binding near the central channel, and again, with either a PEG or lipid-like molecule bound within the central channel. And we just thought, well, maybe the easiest way to go about this is just taking our original hits, taking some lipid-like moiety, and then just sticking them on to the fragments and developing these lipidated analogs that we have right here. And while we're developing the crystallization system currently to allow us to soak these compounds into the APO refolded protein to specifically guide our structural analysis, We've also been playing around with things such as structural analysis tools that are available, such as Chai A1 and Boltz1, to co-fold the protein with these molecules bound to see if they bind within the central channel and mimic some of the lipid-like binding capabilities that we see in our experimental data. And fortunately, what we're seeing is that, at least in Chai-A1, is that for some of these lipid-like analogs that we're developing, we are able to see them binding into the central channel in our prediction models. And of course, we'll have a little bit more about this to come, I think, in the future as we're developing crystallization systems for the refolded protein to do more high throughput soaking into the central channel, and ultimately leveraging those tools to learn a little bit more about how lipid binding biases the equilibrium in the cells, but also what role lipid binding plays for ORF9b. And then, so kind of just bringing this back together, why are we interested in the system? We know that ORF9b doesn't directly bind to Tom70 in this dimeric conformation, but that it has to establish an equilibrium between monomers and dimers where the ORF9b monomers bind to Tom70, resulting in this suppression of interferon response. And ultimately, what we think is that when lipids are bound to the homodimer, there is a tight stabilization of this fold that then prevents it or slowly inhibits the ability for it to establish this equilibrium that then has a consequence on its ability to bind to Tom70. And what we're ultimately interested in finding is some way of mimicking or improving upon that lipid binding capability using a small molecule that can either slow or fully inhibit ORF9b's ability to dissociate into monomers and subsequently bind to Tom70. And of course, I just want to thank some of the great people that have been involved in this. Again, as I mentioned, this is not necessarily a core project within AViD at UCSF. It's actually a developmental project that has stemmed out of AViD's work. And I would, of course, just like to thank all the people that have been involved in both the work that we've been doing and also the work that we've recently published. And yes, that's all. I'm happy to take any questions that you might have.